Good afternoon. I'm happy to introduce the next session, which is organized by Scanbalt, and it's one of the events in the series of the Scanbalt 25 anniversary events. Scanbalt is one of the oldest, perhaps, meta clusters in life sciences in Baltic Sea region, which was founded after the Öresund Bridge was opened in 2000, and their main aim was to connect both sides of the Baltic Sea these times. And one of the main slogan was to have a competition instead of competition, and also try to promote brain circulation instead of brain drain, which was very active these times. Or problems, one of the main problems, what was articulated by many players. And also one main sort of aim was in the previous session as well, that Europe is very sort of not well connected, also when you a perception from outside, and one of the aim was to actually bring together different players to be competitive in a global scene with different big clusters like East Coast or West Coast in US, or Cambridge, Oxford region. So this was the main consideration when this organization was founded. So this today's event is also to actually emphasize strengths what we have here around the Baltic Sea, and also to look to the future. You might see why this painting here is. So it brings us back to the ancient Greek, where the story of Eos and the Trojan prince Titynos, which Eos fell in love with him, and he just asked permission from Zeus to actually make the prince immortal. But what she forgot to ask was to keep him young. And after some time, the young and handsome prince became old and fragile, so she actually left him. So this means that it's not important only to live long or live longer, but to have a more quality in your life years. So, and that's why we are talking about how to extend the actual healthy life years. So here we have some figures about the Baltic Sea region, sort of life expectancy, but we see that there is very different share, which part or the share of the life which is lived average as a healthy life. So, and today's discussions, we see that how prevention and personalized prevention could actually improve this, and to add life to years, not years to life simply. And why we are talking this in the Nordics and in our region, Baltic Sea region. So this is actually latest last year's report on the data on the digital health. And you see that the top of Europe here is two countries, what are represented also today here, Estonia and Denmark, which is considered to have the highest sort of ranking in the e-health index in Europe, according to the last year's report. And the difference is it's not very substantial, but still quite a bit. Today we have representatives in a number of countries around Baltic Sea. We have a keynote from Andres Metspalu. He is a very known person in personalized medicine application and genomics data and population genomics. We have Dr. Krista Kruv from Estonia presenting the real life cases, how to implement the personalized prevention and the sort of population level. We have Professor Markus Perola on Finnish example, Dr. Ulrik Christopher Stolze from Denmark, and Florian Meissner, which is covering the early detection of Alzheimer's. So with further ado, I just ask Professor Metspalu to actually take the floor and start with a keynote.
Okay, thank you. Okay, thank you very much. I see the number of speakers equals this number of audience, so this is quite unique. So let's go. So my title was Personal Medicine and Biobanking and how we reached to from the one to the second. But you know why we need the personal medicine is that we have this. I came from rare disease field like 30 plus years ago, and when science advanced and we had these SNP maps and everything, and it was clear that we can apply genetics also in common complex disease, not only the rare disease. And the concept is that, of course, we have too many. We're getting older, we get more disease, and we also don't have enough doctors. Across Europe, I see the number of doctors are always a problem. The money is always a problem. There's not enough money to treat everybody, and the queues are getting longer. So bottom line is that we have too many patients. And why do we have too many patients? Because we are not applying proper disease early detection and prevention. So if you think about, and this is one thing I did a long time ago, it was starting the newborn screening in Estonia. So you prevent disease if you just detect early the PKU, for instance, and there is a treatment, and all over these years we have probably more than 100 kids saved using this method, just newborn screening. Very easy, cheap, and these kids are growing up, going to school, and live like normal. Otherwise, they just went to the special care homes. So this is everything. We have to move from reactive medicine more to preventive medicine. And I guess this is what I am working. I've been working for the last 25 years. So, of course, one tool to do it was just to have to use a biobanks, because you have to get the population information of variant frequencies from population, the people who are not yet sick. And then you can compare the patients with disease, with controls, without disease, and you can figure out. And there are lots of good biobanks, and I would say the UK biobank is probably the best ones. Most people are using it. FinGen, also 500,000 people. And there are, of course, big banks from US and China. Also, Estonia has a biobank, and there's a good biobank also Qatar. And Qatar is a tiny country, but they have also money they need. And not only Qatar, now the other Arabic countries also doing the same thing. So Estonian Biobank, we started now almost like more than 25 years ago, and we have more than 200,000 people, and this means about 20% of adult population, which allows us to do lots of different research projects, different designs. And we have collected information because everything is digital, like Jaanus was saying. We get annual upgrades from different databases, and the longitudinal approach gives us most fresh data. And we also have genomic data. Everybody's is genotyped, and we have also NMR data from probably 220 different small molecules. And we are open for collaboration. We collaborate with everybody. In our case, tools come to data. Data are not leaving. Samples are not leaving. Samples sometimes still go, but not anymore recent years. But at the beginning, sometimes we had the collaboration projects. But basically, everything can be done in our safe servers, and you only take your own data and leave everything behind. So when we started, the first thing we did was a law, Human Gene Research Act. Served us very well for 25 years, including there was one paragraph saying that government is paying for the maintenance of the biobank. This was a real big relief, and you just don't have to worry where to get the money to buy liquid nitrogen or pay for the energy bills. So finally, it took some time, but from 2007, I guess we have this point in the law. And even now, if we change it, this one stayed. But what was major change is that research data, which are biobank is university institution now, and it was designed for research, not for healthcare. And universities are actually not providing healthcare. We do research and educate people. So that means now this 200,000 people with all these genotypes, and we can calculate the PRS and everything on them. This can be used in healthcare also. So your government don't have to do it second time just to do all this analysis. So this is the main thing. It took quite long time to get all this approved, but now it's from the April 1st, it's a law. So of course, we are collecting tons of omics data, but what I'm extremely proud is that we got funded to sequence long read sequencing, which, if you do the Illumina sequencing short reads, about 10% of DNA, genomic DNA is not sequenced, basically. But with long reads, this improves, and already we see benefits. It's not really finished. Probably it takes still a few months to, I guess we have 3 or 4 PacBio machines running every day. But already we see how clinical geneticists really like this technique, because they can now reanalyze existing material which didn't give them results. But now with this technique, they see some variants and can actually put the diagnosis. So this is European genetic map. It's very old slide, but why we did it? Because when we started in Estonia this project, Europeans said, look, guys, you are in Estonia some kind of a genetic isolate. Nobody is interested, and whatever you do and discover is not important. But, it turns out that we are not so isolated. We are all in Europe, it's just related. So this is, you see on the top, there is a Finns, and the very top is a Finnish isolate even. I guess this was Kuusamo or something. And then it's a big part which is the Baltic countries, Russia, Poland, because in our country we have 25% of Russian speakers. And the Russians across the border, I would say old Finno-Ugric people, also speaking Russian and behaving. But and then it's a big part which is Western and Central Europe. And Italy is on the left corner. They are from Calabria, which is just next to the Africa. So this, if you just put this onto the global map with African people and Japan and China, then Europeans are just one spot. So that means if you zoom in, we see the differences, but if you just compare with the rest of the world, we are similar, very similar. So what we are, I absolutely had no idea who is going to listen, but always the question is, if you do this complex genetics, what do you actually measure? You get this DNA, and what we are measuring is, we are measuring variant frequencies. And you see there is called SNP, which is single nucleotide polymorphism. And you see in one person it's A, and the other one is G, and third one is T. And this is all what we can measure. And this is all why you are all different in certain ways, and we are also similar. And that's why we are all unique. So this is thing. Okay, if we just get the genomic DNA, we do, what do? We probably do like 700,000-800,000 variants from genome, carefully selected, which are most polymorphic, or we sequence the whole thing. But then we compare healthy people with patients, compare the frequency of the variants, and it turns out that some variants are more frequent in cases, or let's say patients, than controls. And this gives us an idea in which region in genome we should look for the variant which is causative, because SNP is not causative, it's not changing, it's just like a milestone on the road. Okay, if we have this information, then there is a new technique or new method which we can calculate the risks. If you have a disease, people with certain variants, and there are many variants which really contribute very small fraction for the full phenotype. Thousands of different variants actually contribute together a big thing. It's almost the same risk as rare disease mutation. Let's say on this, if you do this, you'd get standard distribution. And you go to the far right, you see there is if you take 2%, 1% or 5%, what you take, these are some people who have a highest risk of this certain particular disease. And if you just plot them on polygenic risk score, you see that there are, if you take a top 1%, it's probably the frequency is, the disease is very high. It's comparable. If you take breast cancer, it's the BRCA mutations are almost the same as the top 1% polygenic risk score. So now, how to use it? So it's a new concept. Medical school only recently started to teach genomics in this way as we know it today. When I was in medical school, we had nothing like this. So and doctors are using already also for risk management certain scores. For instance, cardiovascular disease, cardiologists are using a method called SCORE2. And it uses blood pressure, use lipid concentration, cholesterol, so your age, sex, and all this stuff, smoking, and you get the risk. But now if you, and this is what they know, and it's kind of standard procedure. But if you apply now polygenic risk score on top of it, then you see that this group of risk people actually can divide it into three. Some people go up and you get high risk. Some will stay the same, the medium risk. And then some people are getting very low risk. So that means to me that actually if your risk is very low, you don't have to do anything. You just, because your risk is less than population average, and you don't have to apply preventive methods. But now if you just take this score, you apply all this method to all this full group. And this very high risk, you have to do probably very different approach. You have a more aggressive treatment. You have to do immediately something. So that means this is more informative than currently existing practice. And because it builds on the existing method, it's also better adapted. So doctors are more used to it, and you just only increase a little bit. If you start from the beginning, from this like a carte blanche, then it's difficult to educate them everybody. So this is, I believe, it makes sense. And there are two groups. This is on the right side, you see the 60 plus, it's basically the same thing. So this is, if you take top 2%, which is, which gives you, if you see the top curve, the blue one, and compared to the bottom curve, it's the difference is rather big, and this is important, and this can be used in healthcare and should be used. It also women, you see the difference. Also, women are a little bit more protected from coronary artery disease compared to men, but still you see the difference. Okay, this is cardiovascular disease. So let's take another disease, which is Parkinson's disease, also nasty disease. You will hear a story about Alzheimer later, but Parkinson's is also a nasty disease. And it starts kind of a long time before you get the symptoms, but you don't know it. And you see, these are numbers, incidences, 10, 20 people per 100,000. But Germans actually developed a method using ultrasound scanning of substantia nigra, which is a part of the brain in that center. And this is, you see the image. And what it happens in this, when Parkinson's starts, dopaminergic neurons starting to die. And they are dying, and they are dying, and goes year, and 5 years, and 10 years. And finally, 60% or so are dead. Then you start seeing symptoms. Not only the motoric symptoms, but cognitive symptoms, and there are other ones. But all this process starts 10, 20 years before you get the symptoms. Now, if you take, and the idea was that we just take, we had to test now how well PRS works on these ones. We took high PRS people from biobank and low PRS 100 and did ultrasound test on everyone. And you see, of course, neurologists did it. You see their newly diagnosed cases. So people are coming with, and nobody had a disease. Nobody had a diagnosed. But if it just came to the office of neurologist, 9 people got immediately diagnosed. So they were running around, not knowing that they have Parkinson's disease. But and from a control group, only 3. So it's it never works like 100% because there are other causes of Parkinson also. But what was also impressive is that if you just compare the image, and there is a kind of cutoff. If you just have this cutoff, then you see that almost 46% of patients had signals stronger than the cutoff signal. So this tells us that we can do ultrasound to high PRS people because you can't do ultrasound on everybody, but array analysis you can do on everybody because this is so easy, cheap, and you can do it once in your lifetime, and you can use the data the rest of your days. So the idea is that just you use a PRS as a first test, and then invite to the ultrasound the high PRS people, and you see the signal. That's in this case every second was actually future patient. You can start with preventive measures. So of course the question is, we don't have a really good drug for Parkinson, but lots of companies are working on new drugs, and probably the news are also coming. But even simple change of lifestyle, let's say, you have to do more exercise, you have to be more socially active, you have to use your brain more. And actually, there are solid evidence that you can postpone your disease. And if you postpone for years, probably new drug is coming, and then you know that you need it. So I guess this is also very useful, because if people know that there is a risk, they are more ready to change the lifestyle. So we published it. So what we have to really think not only the people in the biobank, we have to actually think about the whole population. But in biobank, we group of basically it came from GDPR basically, because in GDPR it requires that people know what we are doing on the samples and what we are analyzing everything home. So we did a portal called My Genome portal. So and then we calculated certain phenotypes, polygenic risk score, and everybody could go to the biobank website. Like you go to the bank, there's a special secure way to go and see your own data, and this is what we had. We had cumulative risk for type 2 diabetes. We had coronary artery disease. We had pharmacogenetics reports, few drugs, and then we had caffeine metabolism, which people really recognize that I can't drink coffee after afternoon. And so this turns out that they don't really have a second allele and the metabolism is very slow. But there other people are telling, look, I can drink 5 cups per day and still go to sleep and no problem. They had very active metabolism of caffeine. And ancestry reports, so also kind of crude information where you're coming from. And of course, also people figured out that how many percent of the DNA came from Neanderthal, 2%. Most everyone had. But this was also very interesting for people because they had never thought about it. So it's this is something which is unique because usually people don't get information out of biobank directly. So I have to really tell that the EU is also supporting quite a bit now prevention and personal medicine, and there is one big project already from 2018, I guess, 1+ million genome, and of course we still don't have 1 million genome sequence, but if you just collect the clinical genomes and everything else, you're pretty close. So there is a special small project, smaller, is targeting 100,000 new genomes, which is called Genome of Europe. And this is underway, almost 50% is done. And this purpose of this Genome of Europe is that to just get a European database of frequencies, variants based on sequence. We have not only 27 member countries, but we have about 40 different populations from Europe. Let's say in Estonia, you have to sequence all the Russians. In Germany, they have to sequence Turks. And in Netherlands, they have their own colonial history. They have to sequence. So basically, we get a pretty good view what's are the variant frequencies in Europe. And this is for imputation of arrays also can be used because you do array. If you impute it, you get like a sequence which is useful for PRS. Of course, you miss new variants, you miss very rare variants, but it's still very useful. So my vision is that what we have to do in Estonian population and the rest is that we have to sequence certain number of people, get this information for the array imputations and to run SNP arrays on everyone. If you just do now SNP array costs 10 euros. And you do it once in your lifetime and you can use this information all the time for using different algorithms to calculate different PRSs. And I guess this is I call, a population scale personal prevention. And I guess this is what is probably we propose also for Europe was the next funding period. So to conclude, I think that population biobanks were very useful and still are to do this type of research. But I think we also should move from biobanks for population scale because now information technology is so everything is digital. genomic analysis, including sequencing. Now, low-pass sequencing is less than 50 euros. So arrays are 10 euros. Of course, it's 10 arrays, 10, but still you have to get this saliva kit, and so it adds up probably to 50. But still, you do it once in lifetime and compare the healthcare expenditure, so it's not a big money. So it's this is a way to change medicine so that we just move from treating disease more to prevent disease. And this is the whole thing. We increase the years people are living without disease. If we just move from 60 to 65, already big thing. Probably we can move even more. It's not like you just live longer, life expectancy is 80 plus, but last 20 years you are sick. You know, it's not fun. So basically, we should do better, and people are expecting us, scientists and medical doctors, to do better. Thank you very much. Oh, one more thing. Oh, sorry, I missed one slide. Okay, this is the last one. I wanted to show the postal stamp. You know, Estonian Post published a stamp for 200,000 gene donors. So we just can send a letter, put a stamp, which is very nice and colorful, probably in our website. Took some time to convince them, and finally came out very nicely. But somehow I missed it here. Okay, thank you.
Take questions at the end of, if we actually have had all presentations, so be prepared.
Good afternoon. I'm really happy to be here today. It's my first time in Göteborg. It's very cold here, even colder than in Estonia. But I have a pleasure to introduce today one really practical service or example how we have going to use genetic data. And this is, of course, only the first service or pilot phase. And in our pipeline, there are also other services. But let's start with personalized breast cancer prevention. Already some years ago, international community gave a statement, and they came to conclusion that it's not right to keep going like we are doing, we have done so far. So there is a need for certification of population and provide services based of individuals or groups risks. And after that, and also with supporting environment, it was possible to focus pretty fast with personalized services development. And the first of them was breast cancer screening we started to work with. And of course, I should mention that now looking at or coming back to first presentation, then we should in Estonia to catch up with Swedes, because you have much longer life expectancy than we do in Estonia. But now we have to use some smart solutions, and this is one we are going to implement. And the problem is that so-called gold standard has been many years to screen, start breast cancer screening for when women get 50 years ago, 50 years old. And some years ago, there was a change, and some countries started to bring it screening time earlier. And I should mention that Sweden is here a really pioneering country. You have already screening starting from age 40 for some years. But the problem is that starting earlier is not more precision way of screening. And there is a key gap. that lowered age thresholds do not improve precision, and it's not today maybe available, but it's not necessary to screen women with very low risks. And the solution is risk-based screening instead of age-only screening. And Professor Metspalv also already mentioned about all this genetics, but if something is like a knowledge to take home, I think it's that if you are not working with genetic information and data every day, then one good thing to remember is that if we are talking about genetic risks, then it's good to divide them to two. And this risk comes from rare monogenic variants. They are known already quite long, and these variants have really high impact on development of disease, but they are very rare. And there are small variants independently. They have very low risk, but we can summarize them, and then we can put together risk scores, and they give quite the same risks compared with moderate monogenic variants. And if we put this information to Estonian population, then we can see that if we take 100 women, then the pink color women are women with monogenic mutations, and the yellow color women, they are having a higher polygenic breast cancer risk. And as I told, the biggest impact to disease development have these BRCA genes, but polygenic risk could be as high as and the same impact as moderate monogenic variants. And how we progressed in our journey and what we have done to achieve this readiness for implementing this service. Actually, it was a long way, and it shouldn't be so long. But we started with infrastructure and data research already 2018. Then we worked with system implementation, and after that, only in 2019, we started, or 2021, we started in real world clinical studies. And we did it also with our colleagues from Norway and our colleagues with Uppsala and also from Portugal, Lisbon. And the last one we finished or started and finished also, it was last year, the final implementation study in Estonian population. And then it was mainly a technical test of technical environment and also maybe clinical staff who are supposed to give additional information and consult these women. And also year by year, this clinical evidence in randomized clinical studies have evolved. And today we can say that we are ready to start with this service. And we designed in the beginning a really very simplified service to be really sure that we can implement it. So we invite healthy women at age 40 and based on breast cancer polygenic risk, we divide them to two groups. And based on this risk, we provide them mammographies biannually. And these women who have lower risk, they continue as before. They start at age 50. We also involve, of course, familial risk assessment, and we use questionnaire and then geneticist consultation and monogenic pathogenic variants testing, like it's already in everyday use in Estonia, in Sweden, everywhere. And of course, I jump back again to this old law, as I was also working with the establishment of this first Estonian biobank. And I can look back and I can say that it was really important thing to put into this law the possibility to go back to patients or healthy people, gene donors. It was quite a pioneering thing that time, and we participated quite at quite many international conferences on ethics and so on. But today we can say that it was a really right step to put it there. And this year, the new version was implemented, so it gives us possibility to take this data and bring them to our health information database and start to use them in new services. And the other thing that makes it possible is our quite well-developed e-health ecosystem. It's highly connected. And I even don't know sometimes now if I'm working in health insurance fund, if it's good or bad, because if you want to do any development, it takes huge time. It's really a many layered system, and everything is possible, but everything takes time. And in our service pathway, this is supported by a structured step-by-step guidance in the health portal. So it's appropriate to say that all this involvement and information and support and guidance about the service is in the digital way. And the woman who is participating in this screening is guided through this system. All this like waiting times, consents and everything is in digital way. And we involve in our service women genetic data from we have to get new people data and also gene donors data. So we have to bring it all together. And if to underscore something, then maybe now a central system, digital system, in our health information system, the medical device making calculations of risk scores is also implemented in this central system. So we brought all these like small pieces very close to health records, so we can involve more data quite easily. And in our tests, it all works. We will see it pretty soon as our testing period starts maybe in 1 month. So currently we have a procurement for getting for purchasing this medical device. And usually people ask that how do you give answers or feedback to women or results who are participating? Of course, we have two types of medical documentation. One is like official medical report on length standard, but the other one is nicely designed and quite simplified versions we give to women. So we have to say everything that women usually maybe ask in appointment. We try to say it also in this feedback information. And on that report, she knows about her risk, screening recommendations, mammography invitation, time and how she will get it, counseling and familial cancer risk assessment also. And of course, which is important, that we encourage these women to pay attention to body awareness. And we have to claim that if we get any new information, we come back to you and we give you additional information, because this area is developing, and maybe next year or in 2 years we have already better information or better service, so we maybe have to go back to them and add some additional information. And I'm going to finish. Our story have been long, and I think that also one reason for this is that Estonia has quite small amount of money compared to other countries we had used to this development, and that's why we made quite many things and developments that other countries are doing as a really big program. We divided it into small projects and tried to put them together and navigated through this all this pathway. So it made it longer, it made it maybe more expensive, but seems that we can pick these fruits in a couple of months, and we plan to launch our service in September. And if to say something as a take home message or something like this, then my experience is that if it's really such a complex system and development, it's good to go as fast as possible to real clinical environment. Because if you are doing your projects and developments somewhere in lab or semi lab, then it every time you go then to real environment, then you see that we need to change something, and it's more expensive, and then it triggers again the next round and round. So maybe this is the one and the most important message. Thank you.
Good afternoon. Good afternoon. I'm Markus Perala from Helsinki, Finland. I'm going to go to a bit back in steps here, how we identify the people for personalized prevention. In Finland, we have two large population-based studies. Another one is about 10% of the adult population called FinGen, where we do genome-wide association mapping, and then a Fin registry where we have the whole population. And it will describe how we find out risk, how we found risk factors for various common disorders here. So FinGen, as I said, it's almost 10% of the population. It was started in 2019, and currently we have performed all the genotyping. We have genotype about 521,000 ethnically Finnish people, and somewhat more if you include the foreign residents or foreign-based residents in Finland. And we, as I said, we don't sequence anyone, but we do the genome-wide association chip, so-called FinChip on them, and then we map this against the mostly registry-based data from Finnish health registers. It's so-called genome phenome study. So we study all the total genome against all the phenotypes we can derive from population registers that have more than 100 people as endpoints. Currently, we have about 400 to 500 disease endpoints that we do GWAS every year with added data and about 20 million variants from these imputed chips. This is the PI of the study is Professor Arno Palotia, whose vision led to this project. We are now at phase 3, and we are now planning phase 4 starting 2027. The first 3 phases were partly funded by Finnish government, and then 14 different pharma companies. So this is a public-private partnership, pre-competitive, and we have more than 1,000 researchers from Finland and these other partner organizations doing the studies. The core analytical group is based in Finland and has about 20 statisticians, bioinformaticians, and physicians, led by Professor Samuli Ripatti. Currently, we have more than 3,000 disease associations and well more than 3,000 disease endpoints analyzed. Of these, 400 were Finnish-specific associations. As Professor Metspalu said earlier, the Estonians are close to Europeans. We Finns are genetically isolated, which is a bonus in that sense that we can find new variants and even new genes for various diseases. I'll show some examples about that associate with the diseases as compared to other populations. That we have actually more of these rare variants in proportion than less isolated populations. However, we still are Europeans, so most of the GWAS peaks that we get do replicate in other European populations and vice versa. So what I'm really excited is that we have actually identified truly protective variants. Of course, those are more rare, but before this project, I really thought about protective variants as like a two sides of the coin. So okay, you don't have the risk variant, so then your variant is protective. But now we have truly found functionally some variants for different diseases that do seem to have protective effect. So we have to, like, this complicates the risk calculation, but it of course tells us a lot about this disease physiology, and more than 1,000 publications in this far. Including the first that I know of, the cover figure for Nature, this paper written by Dr. Mitja Kurki, who's one of the chief bioinformaticians in the project, actually leading the Broad Institute team with Mark Daly for the analysis. This was published a couple of years ago in Nature. It's a nice paper if you want to read more about FinGen. Pretty much of that still holds true, although now we have much more data on proteomics and other omics as well. So some of these novel findings that we have are that, like, we found a Finnish-specific locus for inflammatory bowel disease that actually seems to associate with more severe progression of the disease, and it's not the rare variant in Finland. It is in single digits, but anyway, thousands of Finns carry it. Then we have actually found a variant in ANGPTL7 gene, which protects from glaucoma, and we have measured the eye pressure in people who carry this, and they have actually lower pressure, although they don't yet have glaucoma, and probably never will be. There is protective variant for coronary heart disease is MGFE8, which actually affects the contractility of these vessels, etc. So these are truly protective variants, which were not very often shown before this kind of large-scale analysis. Of course, we have found a lot of predisposing variants as well. So, for example, here's the example about the Finnish specific variant that protects from glaucoma. This is not seen. Some of these we do collaborate with our Estonian friends, and you try to replicate their results, or they replicate our results, and some of these are seen in Estonians, but many of these 150 are only seen in Finns, and as they tell that it's not only bad to have it isolated, you get new ideas about the disease pathophysiology from that kind of data. Then that was the FinGen. Then I will go to the Fin registry, which is THL. I work in THL. This is a project I'm the PI of. And here what we did, we have access to our institute, which in our institute it's our law-abiding task to analyze the registry data. So it's told in the law for THL that we have to do this analysis. So it was pretty easy for us to get the permission. We have now here joined together 19 different registers from earliest in 1950s. Which has resulted in 7 billion lines of data, and so 7,000 million lines of data, including, for example, all the laboratory results from that have been done on Finns since 5 years ago. But also much older people, older data as well. As I said, for cohort purposes, we selected here all the people that were alive in 1st of January 2010. And so we have more people in the study actually that we have now living in Finland. In Finland's population is about 5.53 million people, but we have about 7 million people in the 7 billion line data set. So if we are much larger than the FinGen, of course, we don't have the genetic data here. We actually can't join this to the we don't have permission to join this FinGen data. However, FinGen now in its phase 3 is encouraged by the studies we have done in Fin registry, is aiming to do the same as we will do after 2027, join all this registry data to the genetic data they have. I work also in FinGen, so I should maybe say we have. So in Fin registry, we analyzed the risk trajectories of disease and build outcome-specific prediction models. We use also machine learning and deep neural networks in this analysis. So do some kind of a hypothesis-free analysis here as well. And we try to bring new epidemiological and biological hypotheses and have prevalence and distribution of the diseases. We have actually built a website called Risteys, where anybody in the audience here or in Finland can go and look for her or his favorite disease by ICD codes and see its prevalence in Finland, age-related. Then I'll just go through a couple of examples of the power of Fin registry, why I am so excited to have this. We have more than 10 different projects now going within Fin registry. My naive thought was, okay, we will just do one huge analysis and analyze risk factors or all of these 4,500. We have the same phenotype system as in FinGen, so we have about 4,500 different phenotypes we analyze, but that was too heavy analysis to comprehend and also to process by our current computing power, although we do have access to the Lumi computer in northern Finland, which is one of the better computers in the world. This was done already during the COVID time. We wanted to see, our institute is also responsible for vaccinations in Finland. We wanted to see where to direct these prevention called vaccination and who doesn't get vaccinated. And we selected the first vaccination available in the first year of the pandemic, that who came and took because it was voluntary for anyone else than but healthcare workers like myself, we had to take the vaccination to work, but for other people it was totally voluntary. And what we did, we used these 19 different registers and made a machine learning model using XGBoost type of machine learning and tried to pinpoint exactly, not taking any hypothesis, which things prevented people from taking the first vaccination. So this is a Shapley plot that we got from that, and actually interesting was that the most important thing for not taking the vaccination was the income, and we had two variables, and they were both most important. The more important of these two income variables was that if you didn't have any income in the year of 2018, then you didn't take the vaccination more probably. And the second one was actually the quantitative value. So the less you earned, the more prone you were to take the first vaccination, which immediately directed us to the socioeconomic status, that we knew that, okay, somehow our message is not reaching these people who don't have a good income. Then some other things like drug purchase, disease diagnosis actually worked the other way around. If people had this doesn't appreciate whether it's a risk factor or protective factor for taking the vaccination. So if you have diseases, you are more prone to take the vaccination, etc. Here you can see that if you ever receive social benefits, that was a risk factor, and also if your mother tongue was other than Finnish or Swedish, and this again targeted our prevention people for going into the immigration groups. Of course, these are kind of if you think about obvious things, the low-income immigrants, the message doesn't reach them very well. But it was important to do it scientifically, to be objective, and not to let your, own thinking be the only source of these decisions. Also politically, it was important. And then some other things like different mental diseases, substance abuse. Also prevented were like preventive factors for getting the first vaccination. Also, whether your first relative took or didn't take, especially didn't take the vaccination, was again indication that you were not so prone to take the vaccination. All these odds ratio were not so big, but when we built a risk score, not this time in genetics, but about these different risk factors, you can of course do it with these. Actually, 1%, the highest risk, and 1% out of 5 million people it's a lot of people. So their vaccination rate was only less than 20%. So I think we were really successfully in showing the proportion of people in Finland who don't want to go to vaccination. I don't know if we delivered this information to the people who were responsible for vaccinations, and I hope they applied that. Whether they succeeded or not, that data I don't yet have. We will follow this up, of course, but I haven't yet done this. Then another thing, what we did, and this is also example that even if you think you have registry data, you think that, okay, now I have all the data, it's perfect, now I can do decision based on science. Well, sadly, that is not always so. And this showed this as well. So we again took this whole population sample and did some deep learning analysis on the 1 year mortality. So can we predict who dies within a year? And if we consider the 5% of individuals that, given our risk factors, I'll come back to this shortly, the mortality rate was almost 17 times more than 1% average. And we did capture in our risk models more than 70% of all the deaths by the registry data only. So again, here's no genetic data here, just the ones. And we also could predict well the hospitalization risk and annual drug expenditure about these risk factors. And actually this data set was the same that I showed for the vaccination for different registry-based risk factors. What we found out that this model was not equally predictive in all Finnish regions. So there were the darker regions where this worked well, and the lighter regions is where they didn't work very well. The two white spots on the bottom is we didn't have data from them for some reasons, or not. So they are probably not so bad. But anyway, these lightly colored the data didn't really work so well for them. And also when we dug deeply in this, again, the income. So if you're doing not so well socially or in life, if you're low income, you don't really see a doctor a lot. If you're like have a drug problem, if you're mentally ill, you don't maybe have the resources, mental or financial, to go to a doctor. So you don't get these registry notifications, meaning that even registry data, also it is population based, is not a totally fair, totally objective interpretation of the population health. Of course, this is saying it very boldly. Of course, it does work pretty well for most of us, but there are a significant proportion of people who are not lighting up in these registry studies, and those are people who don't go and visit the doctor, the healthcare so often, and for many reasons. And so, as I said, so it's less predictive in individuals belonging to multiple sensitive protective groups. And now this is very exciting. We just got about a couple of years ago all the lab results since 2014. We don't have all from 2014. We have all from about 2021. So if you have gone to a laboratory in Finland to measure cholesterol, we have it, and we can use it for predictive models. This is about 1 billion lines of data, and it's a big data set, and I don't have really anything to show you yet, but I'm sure in a couple of years we will have some exciting results, and maybe I'll come back to here talk more about this. And I'll just finish here. This is just some general statements about Finnish registry and FinGen. In Finland, we have the luxury that people trust us scientists. They want to participate in these studies, and we have published this Finnish registry very widely in media. And out of 5.5 million living people, 50 people, 50, have said that they don't want to participate in this. Of course, you have to be active to say it, but this has been very widely in media. So I just want to end by thanking the Finnish people for letting us do this. This is very important and exciting. Thank you.
All right. Can you hear me? Is it on? No, now it's on. Thank you. All right. Well, my name is Ulrich Stelza, and I'll be talking a little bit about newborn sequencing and screening and how we do it in Denmark, and then about the Helix project that we're planning. And I wanted to start with this little girl. She's 3 months old. Her name is Danielle, and she has tyrosinemia. That means she has a lot of tyrosine in her blood, and this is caused by a downstream effect. So she's missing an enzyme that's supposed to take one molecule and push it up this line, and basically everything from there builds up. And that's unfortunate because one of these compounds can then change into a toxic form, and this will create liver toxicity, and it will create organ failure, and ultimately it will result in death for this patient. And the only way to really fix it is to do dietary restrictions and then to do a liver transplant. And doing a liver transplant in a 3-month-old is really not is high mortality and very problematic. So this is where the flower that was in the first picture comes in. So this is the bottle brush plant. It looks like a brush you use to clean a bottle. And some researchers right here in Gothenburg, and this is why I included it, found out that there's a compound here called now nitisinone that can prevent this from occurring. So it blocks an upstream enzyme, and this removes this toxic chemical, and it alleviates the patient from needing this liver transplant. So today we're screening for tyrosinemia in all newborns in Sweden, Denmark, and many, many countries around the world. So millions of babies are going through this every single year, and the researchers published this in the Lancet in '92, so this is an old story. This was one of the first benefactors of this particular drug, and she's now working as a nurse. So she's had a long, very productive life. So this began way back in the 70s in Denmark with some really ingenious methods that we no longer use to screen for these first 2 diseases. And then there was sort of a dark period where not a lot happened. And then in 2002, we started expanding the newborn screening. So this was using new methods, mostly mass spec, but also other assays, and we slowly introduced more and more conditions on the list so that there are now 25 diseases that all newborns in Denmark are being screened for, and these are the modalities now used to screen for these conditions. So if you took some time and read through this, you would see that pretty much all of the conditions have genetic causes, and despite this, we're using biochemistry to detect them and not sequencing. So we're not doing any sort of sequencing to detect these diseases. These also, so because we're using biochemistry to detect them, we're also looking for a certain type of disease. So a lot of these diseases fit onto the map of human metabolism, that if you've gone to any sort of medical or biology degree, then you've maybe seen this, and so you can see where things go wrong on this map. And you can also organize it like this, where you're looking at amino acid disorders, organic acid disorders, fatty acid disorders. So these are things you can imagine you can kind of measure, like you measure a pH with a bit more advanced techniques. But then there's endocrine disorders and lysosomal disorders, and then this category of others that we're starting to expand into. So newborn sequencing, as stated here, has really not kept pace with the development in genome sequencing and the therapies that we have available, right? So this goes way beyond, and that means that a lot of countries now are looking into expanding newborn sequencing to really, or newborn screening to include sequencing to look for many, many more diseases. And so we want to jump on the bandwagon, and we're working on this project under the name of Helix, because you use this little blood spot that you do a little needle stick on the heel, and then we want to look at the genetics. And Mette Nygaard was supposed to be here today and is regretful that she couldn't. She sent me instead. I'm assisting her and working on this project, and we're working on a gene list to see what kinds of diseases would you look for if you're gonna do screening of newborns. And so there's hundreds of diseases that we've considered. We're looking at about 100 more that we would like to include in sort of an experimental setup. So this is research and not nationwide implementation. And I just want to show you 5 of them here at the top that are the most agreed upon, and they fit quite nicely onto the previous framework. But for various reasons, there's not a really great biochemical assay to screen for this. So it can be challenging. And you can also put these, if you want, on the map of human metabolism, and they're very similar. So here's one of the patients that can have one of these conditions, and he has what is known as CPS1 deficiency. And it's not super important what the enzyme does, but it is a super important baby. This baby right here is called Kyle, and he was named one of nature's 10 most important people of 2025 in science, along with some Nobel Prize winners and so forth. So it's a big deal. And New York Times wrote just a month ago that this may be the most important medical story of the decade. So what is the story? I'm curious how many people here have heard this most important story of the decade. Well, so Kyle, just like our previous patient, is born with a genetic condition with a metabolic disease, and he has a CPS1 gene that doesn't work, and this causes a disruption to the urea cycle, and so he's on a special diet, and he needs a liver transplant in order to survive. And he's a newborn, and so you really want to wait as long as possible with giving liver transplant, and preferably not give it at all, but there's no treatment, right? So in this study, down here in the corner in New England Journal, they did a Project Warp Speed like we did for the vaccine for COVID, but they did it for this single patient. So in 7 months, they went from idea to cellular testing to animal model to FDA approval to administration to the patient when he was 7 months old. And he was born on August 1st in 2024. And this was a CRISPR-based therapy, but unlike any CRISPR therapies that you may have heard of, this actually fixes the core problem. So this goes in and changes the DNA, has a missense mutation. It changes the the nucleic acid from an A to a T, and then fixes the missense mutation. And then he has a liver that has a functional CPS1 enzyme, and he no longer needs a new one. So here is Kyle, now almost 2 years old, with his same original liver. And the hope is that he can go on using this. He's still on a special diet, but he's using his own liver to process it. And so this is kind of the original, I think, idea of ultra personalized medicine. We have one medicine that was developed just for Kyle here. So that's how it can be maybe used. So we can screen for this and find it early, and maybe use therapies like this in the future for many, many of these diseases. And there are many. This is just an example. But this is sort of traditional newborn screening, looking at these biochemical issues. But there's this category of others, and you could really expand that quite a lot if you start sequencing everybody. So you could look at cancer predisposition, you can look at cardiogenetics, so heart disease predisposition, sensory loss, like inherited vision loss, blindness, or heritable hearing loss and deafness, and then hematology, and the list is quite long. So we don't really have time to go through all of this, so I just wanted to give you some examples and then think about some of the problems with expanding into this new realm. And one of them is using whole genome sequencing. Is it accurate? And what do you do with all these variants of unknown significance? A lot of concern about, is it going to cause overdiagnosis, overtreatment of patients? Is it safe to do it and to store the data? And is it cost effective? I'll give a brief perspective on some of this and skip some of it, I think, in the interest of time, because I want to give Florian a chance to also speak for a while. So we have a lot of EUSs. It's a growing problem, but we have an interesting situation here where we know the true prevalence of all of these diseases that are shown here, because we've been screening for them for decades. So we know exactly how common they are. And so when we look in population databases, like Nomad and UK Biobank, we can actually compare and see the true prevalence and see, based on the genetic prevalence, which is on the x-axis here, how does that compare to the known true prevalence? So that's really not a guess, that's the real prevalence. And they correlate quite well. So even though there's a bunch of VUSs in these genes, if you do a very simple filtration on pathogenic variants, you basically diagnose the number you would expect. So it says a 80% correlation. So I want to show you this example of hereditary retinoblastoma. So in Denmark, we have 60,000 babies every year, and most of them don't develop a childhood eye cancer. So it's really rare. Just 5 of them will develop a retinoblastoma within the first 5 years of life. And we could do screening for this. It's quite simple. You look at the patient's eyes, the child's eyes, with a slit lamp, and ophthalmologists can do this very easily. But you have to do it quite regularly, like every month. So every month, every baby would have to go to the ophthalmologist. So that would be population screening for retinoblastoma, and it would have the benefit of potentially finding these and treating them early, and then you could prevent some morbidity in 5 children, but you would send 60,000 children with their families to screening with the risk of false positives and so forth, and this very quickly becomes a bad idea. So this is not what a geneticist would sort of approach it, but we of course know that the RB1 gene can be mutated and cause hereditary retinoblastoma, and this changes the calculus quite a lot. Most of the children that are born don't have a mutation in this. We expect that about 3 have it every year. But they have a 90% risk of developing retinoblastoma in the first 5 years of life. So this is truly like precision screening if we send these patients to the eye doctor. So now we can prevent morbidity in 3 patients potentially, but the costs of doing it become very, very negligible because you're just sending 3 kids to the doctor. So this shifts the whole balance. Now, there was a study that I was a bit concerned about, still not been published, where they looked through all of UK Biobank and Nomad, and so that's 800,000 adults, and they saw that there were 25 carriers of these RB1 mutations, right? And you would expect them, I just told you, they should have had a eye tumor as a child. The problem was that they didn't. Only 28% of them had this. So so they basically conclude that the penetrance is not what you would assume, and this changes everything for newborn sequencing. You're going to do overdiagnosis, you're going to do overtreatment, send too many kids to the eye doctor. Okay, but we are working on this in across the world. The generation study has already sequenced 20,000 babies. They found baby Freddy. He has an RB1 mutation, and he does have an eye tumor, and he's been treated, and he now has perfect 20/20 vision and has been treated with minimal side effects, right? Still has his eye and everything. It's also the BTI study out of China. Similar story. In total, these are unpublished data. The International Consortium on Newborn Sequencing, they have 5 patients like this, and 3 of them have eye tumors. So the 28% number is not correct based on this data. So maybe it's somewhere in the middle, and we'll learn by actually doing it. Okay, so we have quite a lot of PKU cards in Denmark. We have so I think I'm going to skip this in the interest of time. We have like a quite high number of PKU cards. We have developed a method to screen them for these rare variants like RB1 quite efficiently and cheaply. And that's part of what we're working on, what we want to do. And this is true for a lot of the Nordic countries, with Sweden, Denmark, Iceland, and Estonia being kind of leading the pack on number of cards available. So I will skip data safety. I have some ideas on how to make it more safe that you can maybe try to guess from this. I don't know. But we'll skip it in the interest of time and go to my final thoughts on some of the cost effectiveness of this. So if you look at critically ill newborns, it's extremely costly to treat them. So you take this little person and they need to take care of everything. And so on average, a hospital stay in the US, which is hard to compare to Europe, but costs about 4 million Swedish kroner. And in the US, there are 400,000 babies in the ICU every year. So we're literally talking billions of kroner. And that means that on average, every single child has a huge, we contribute to this quite a lot as a society. So then the final thing here is that genetic conditions contribute substantially to this number, but it remains quite unelucidated. Looking at just childhood cancer that I showed you before, it's been demonstrated in some models that if you can get the cost low enough so you can screen kids very cheaply for this, then it becomes even conservatively cost effective to do it. And that's just looking at childhood cancer. So I think there's a cost effectiveness wise, you can argue that there's interest in looking into this. But that's looking at it very utilitarian. So it's saying, how do we get the most out of this? And so I'll end with these philosophic thoughts with these two Johns. So this John Stuart Mill, who has this utilitarian idea that we should get the most out of every dollar we spend. And I think you could argue from his point of view, but I think it's a much stronger argument if you argue like John Rawls over here. So his argument comes from his theory of justice, that we should try to create a society where everyone has equitable access, right? And we have children that are born with these mutations totally randomly, and it's the most unfair thing I can think of. You're just given this mutation, right? And then we have someone like John Stuart Mill saying it's a bit expensive to fix, right? So if you argue like John Rawls, then you could say, well, maybe as a society we have a duty to help those who are most disadvantaged, and I think this falls maybe in that category. Okay, so we're working on this Helix study, and if you have questions about it, I would love for you to direct them to Mette, who is, like I said, regretting not to be here, and also to me afterwards if you want. Yeah, and then I will pass on the baton to Florian. Thank you very much.
Thank you very much for the presentations and very impressive content today. Now I have to shorten my presentation, and I try to do my presentation in 3 minutes instead of 50 minutes, so it's a very fast situation. Dementia is increasing today from 1.8 million to 2.3 between 2.3 and 2.7 million in 2050. And so we have a problem in capacity, and we have a problem in to care all these people. So our approach is to go on a guideline-based care, which means that we are staging the patient. And staging means letztendlich we beginning with the syndrome detection. Next one, the exclusion of other causes of the injury. And additionally, we using etiological diagnostic and individual care planning. From the first step to the last step, in one circle, it depends 26 months. From the first impression of cognitive impairment to a real diagnosis. It's too much time, and so we have to look how we could shorten the time. So we have a pilot project in Cologne, which is called ALFI. ALFI stands for Alzheimer leitliniengerechte Früherkennung in Köln. So we have an approach to connect physicians, counseling services, patients, and caregivers in one structured patient pathway, and to get the patients letztendlich in a real care management system. But we have not resources letztendlich every patient older than 65 to detect or to screen of dementia or of Alzheimer dementia. So we have to build a gateway to detect patients earlier and cost effective. So we have a cooperation with a company in Germany. It's called KI Elements. And KI Elements has an AI agent, letztendlich, who calls a patient and makes an assessment with memory functions and speech recognition and some things like this. And after this, we get speech biomarkers, which sounds letztendlich very crazy, but the processing speeds, the executive function, and the learning and memory building up one global score, which detects Alzheimer's disease or dementia, and we are able to put this letztendlich in the right situation, in the right stage of a patient. So we could put the right therapy and the right diagnosis to get the patient as fast as possible letztendlich in care. So one type of care we have in Germany is the new prescription called apps called DIGA. So Memodio is one of them, and the first prescription apps for early stage dementia with reimbursement in Germany. And it's built up on cognitive training, exercise, social participation, nutrition, and risk factor optimization. And the outcome is letztendlich patient have a better cognitive functions, better daily abilities, better activity level, and a better life of quality. Every patient uses, or 75% of users letztendlich use Memori every second day. And the top users have this app, using this app more than 2 years. So now it's the question letztendlich how we are coming letztendlich in this situation of care. We have not enough resources to get every patient in these ways. And so our system letztendlich is possible to guide the patient in the real situation and in the pathway. And our system letztendlich supports the pathway, the reference, coordination, and the longitudinal case management. And so the patient is detected after 3 months instead of 26, and the therapy could begin. So this is our ecosystem, and IFI Digital is one solution for Alzheimer's disease. And our partners, KI Elements and Memodio, are integrated, possibly to detect Alzheimer's dementia or to have new therapy programs and so on. So it was the express variant of my presentation, and I hope you have a little impression of my presentation. Thank you very much.
So maybe I would ask the speakers here, because we have online sort of broadcasting of this session. So perhaps it would be better to come to the screen, to the scene, and we can actually have a sort of 25 minutes of discussion, just to take where you first of all, I would ask if there is any questions, any comments from the audience at the spot. Seems no, but maybe later. So I would ask and start with sort of question which will enable you to actually make some kind of foresight. What would you see changing, or what will happen in 5 years in a particular field, what you are working, and what you think in the practical terms will be different, taking in the new advances in the AI, and maybe we have a European health data space which can change the screening of the secondary health data use. Maybe we will start from Krista. You maybe you can say where would you be with risk personalized risk-based services in Estonia, for example.
If I look ahead, like 5 years, maybe probably we would like to bring more genetic data or create more genetic data and reuse them in different services. After the first service which I introduced, then the prostate cancer prevention, pharmacogenetics, cardiovascular diseases, all this information already available, and we can use it. And if we can reuse data, then it makes it really cost efficient also. So that's my.
Okay, I just got a note that our session is end, but let's get a quick tour still, and because I'm mixed up, I thought that we have still some minutes to go. So, but Ulrik, about you?
Oh, I think I think this, like, what I presented is where we're heading. There's no doubt you're going to see more sequencing of newborn. It's already happening, and so that is coming, and we'll see what the implications are in terms of disease prevention, and also how the public reacts to it, and how we use and misuse this data is pretty interesting to follow. Yeah, that's all I have to add.
Anders?
Yeah, in 5 years, I believe that we have made shift from reactive medicine to preventive, in terms that PRS has been accepted as part of the risk formula, so in addition to the environmental and behavioral risks. And I see that AI is taking a much bigger role in communicating these results to people, and also in education, because okay, that's it. Yeah, I see AI and population-based screening using genomics.
Markus?
Yeah, just to say something different is that in 5 years we will have results of 2 very exciting cohort studies of sequencing 100,000 babies, one in UK and one in US, and those results will probably be out by then, and I think that will change the prevention of childhood disease significantly in future when we get these population-based or cohort-based results of these hundreds of thousands of babies. This is, I'm really expecting those results.
Thank you. And Florian?
I think the decade of screening and detection is ending, and we are going into a predictive modeling letztendlich of diseases and impairments. And so it's cost effective letztendlich to hold on the perfect level on quality in care.
Well, thank you so much for your presentations, and sorry for mismanagement of this time.